Welcome to G-Fun Facts Online. Today we are shrinking down to the scale of a speck of dust to explore a universe. We are talking about the brain of the common fruit fly, Drosophila melanogaster. For over 100 years, this tiny creature has been the workhorse of genetics, but inside its cranium lay a biological machine of such staggering complexity that it confounded scientists for a century. We knew the parts list was vast, comprising tens of thousands of neurons and tens of millions of connections, but the schematic was blank. We could see the cities, but not the roads. That era of ignorance ended in October 2024. In a landmark special issue of the journal Nature, a consortium known as FlyWire, led by researchers at Princeton University and the University of Cambridge, unveiled the Connectome Atlas. This is the first complete, synaptic resolution map of the entire adult fruit fly brain. This is not just a map, it is a Rosetta Stone for neuroscience. Comprising 139,255 neurons and 54.5 million synapses, this digital atlas represents a leap in scale so immense that it effectively creates a new discipline. To give you context, before this, the only adult connectome we possessed belonged to the roundworm C. elegans, a creature with a mere 302 neurons. The fly brain is 500 times more complex, capable of navigation, learning, courtship, and combat. As Mala Murthy, the co-leader of the project, said, "The fly is not a reflex machine, it has a mind." Let us walk through the epic journey to map that mind, the revolutionary technology that made it possible, and the startling discoveries that are already rewriting our understanding of how brains perceive, decide, and act. The history of neuroscience has always been limited by the resolution of our tools. We could record the electrical firing of a single neuron, or we could scan the blurry patterns of millions in an fMRI, but we lacked the Middle Earth, the precise wiring diagram that links individual cells into functional circuits. The dream of a whole brain connectome began with the worm in the 1980s. A project that took over a decade of manual labor. To map a fly brain, which is orders of magnitude larger, by hand was calculated to take 50,000 person-years. It was a task that was, practically speaking, impossible. The breakthrough came from an unlikely marriage between cutting-edge artificial intelligence and a global army of citizen scientists. The project began with a single female fly brain, sliced into 7,000 nanometer thin sections and imaged with an electron microscope at the Genelia Research Campus. This generated 21 million images, a data trove so vast it would fill a library of hard drives. Enter Sebastian Suong, a Princeton professor and pioneer in computational connectomics. His lab developed AI algorithms capable of tracing the twisting, turning paths of neurons through these 2D slices to reconstruct them in 3D. But AI, while fast, is not perfect. In a dense tangle of neural spaghetti, the AI would occasionally merge two neurons into one or break a single neuron in half. To fix these errors, the team turned to the FlyWire legacy, a gamified platform where human volunteers could proofread the work done by the AI. This evolved into the FlyWire consortium. While the heavy lifting was done by sophisticated deep learning models, the ground truth was verified by a unique coalition of professional neuroscientists and devoted gamers from around the world. As Sven Dorkenwald, the project's lead author, explained, they built a Google Maps for the brain, but unlike a satellite image, they had to label every single building, street, and alleyway. The result is a publicly accessible database called the FlyWire Codex, where any researcher can log in, type a neuron's ID, and see exactly who it talks to, who talks to it, and what neurotransmitter it likely uses. It is the democratization of brain data. Now, what does a mind look like when you strip away the skull? The Connectome Atlas reveals a structure that is both hierarchical and chaotic, modular yet deeply interconnected. One of the first global statistics to emerge from the map is the existence of a rich club. In network theory, a rich club is a group of high-degree nodes or highly connected hubs that are more densely connected to each other than to the rest of the network. In the fly brain, this club comprises about 30% of the neurons. These are the power brokers of the mind. They do not just process local information, they integrate signals from across the brain. The researchers identified two distinct types of power players within this club. First, the integrators. These are neurons that receive a massive amount of input from diverse sources. They are the listeners, gathering intelligence from vision, smell, and internal states like hunger to form a coherent picture of the world. Second, the broadcasters. These neurons send outputs to a vast array of targets. These are the commanders, disseminating decisions to multiple motor circuits simultaneously to coordinate complex actions. This architecture suggests that the brain is not a linear pipeline, but a bow-tie structure. A wide range of sensory inputs compresses into a dense core of high-level processing, the rich club, which then expands back out into a wide range of motor actions. While the connectome maps neuron-to-neuron links, the projectome maps the highways between brain regions. A standout discovery here was the role of the subesophageal zone, or SEZ. Previously thought of as a mere relay station for taste, the Connectome Atlas reveals the SEZ as the grand central station of motor control. It is the primary hub where the brain talks to the body. Almost all descending neurons, the cables that run from the brain to the nerve cord, which is the fly's equivalent of a spinal cord, pass through or originate in the SEZ. It is the bottleneck through which thought becomes action. Now let us look at the senses. The fly is a visual creature. Its two compound eyes are marvels of engineering, and the Connectome reveals that a staggering 50% of its brain is dedicated to processing vision. The map of the optic lobe alone contains over 50,000 neurons classified into hundreds of cell types. One of the most critical functions of a fly's visual system is not to admire the scenery, but to avoid being swatted. The Connectome has detailed the specific circuits responsible for detecting looming objects, which are dark shapes that expand rapidly in the visual field. The map shows a direct hardwired pathway from specific visual projection neurons to the giant fiber system, a set of massive neurons that trigger the jump and fly escape reflex. This explains why flies are so frustratingly hard to catch. The processing does not need to go through the decision-making centers of the central brain. It is a high-speed bypass, a reflex arc sculpted by millions of years of evolution to prioritize speed over nuance. Beyond the large compound eyes, flies have three simple eyes on top of their heads, called ocelli. For years, their exact function was debated. The Connectome Atlas put the debate to rest by tracing their wiring. The ocelli connect directly to a dedicated set of descending neurons that control the neck and wing muscles. They act as a biological gyroscope. Because they are unfocused and extremely sensitive to light intensity, they can detect the horizon line and rapid changes in body roll faster than the compound eyes. If a gust of wind tips the fly, the ocellular circuit detects the shift in the sky's brightness and automatically adjusts the wings to level the flight path. It is an autopilot system, fully decoded. Perhaps the most exciting aspect of the Connectome Atlas is its ability to explain behavior. By tracing the wires, scientists are finally understanding the mechanics of how a fly walks, stops, and grooms. For example, how does a brain command a body to halt? It turns out, stopping is not just the absence of going. It is an active, multi-layered process. Researchers using the FlyWire data identified three specific types of neurons that control stopping, whimsically named foxglove, bluebell, and brake. Foxglove and bluebell act as the mechanism for taking the foot off the gas. They use the neurotransmitter GABA, the brain's primary inhibitor, to silence the walking initiation circuits. When these neurons fire, the drive to walk is suppressed. However, the brake neurons are different. They use acetylcholine, an excitatory transmitter, to actively stiffen the leg joints. This is the slamming on the brakes mechanism. It locks the posture, ensuring the fly does not just drift to a halt but freezes instantly, which is crucial for a creature that needs to land upside down on a ceiling. Conversely, the drive to move is controlled by neurons dubbed roadrunner. These cells sit at the top of the motor hierarchy. The Connectome shows that roadrunner neurons integrate sensory data, like the smell of food, and internal states, such as hunger, to generate a graded forward signal. The stronger the roadrunner fires, the faster the fly walks. This signal is then demultiplexed by downstream circuits into specific commands for each of the 6 legs, coordinating the tripod gait that keeps the insect stable. Grooming is another behavior that has been decoded. Flies are fastidiously clean, constantly brushing dust off their eyes and wings. The Connectome revealed that this is not a random act. There is a somatotopic map for grooming. This means that the mechanosensory neurons from the eye connect to a specific eye cleaning circuit in the brain, while neurons from the wing connect to a wing cleaning circuit. It is a physical representation of the body within the brain. If you stimulate the eye circuit, the fly will rub its head, even if there is no dust there. The map explains how the fly knows exactly where to scratch. Perhaps the most sci-fi application of the Connectome Atlas is the ability to run the brain on a laptop. In a companion paper, researchers took the wiring diagram of the feeding circuit and built a computer model. They simulated the activation of sugar-sensing neurons and water-sensing neurons. The model successfully predicted, with over 90% accuracy, which downstream neurons would fire. They discovered that the circuits for sugar and water are not separate, parallel lines. They are deeply intertwined, sharing many of the same integrator neurons. This suggests that the fly brain processes reward, which is sugar, and thirst relief, which is water, using a shared currency of value, calculating the utility of a resource based on the fly's current need. This success proves a fundamental hypothesis of neuroscience. Structure predicts function. If you know the wiring, you can predict the thought. We are entering an era where we can debug a brain just as we debug software. Finally, the completion of the FlyWire project forces us to confront deep philosophical questions. If a mind, even a fly's mind, can be fully mapped, digitized, and simulated, what does that say about the nature of consciousness? The fly brain is determined by its connections. Every courtship song, every escape maneuver, Every decision to eat or fast is the product of physical wires carrying chemical signals. However, the snowflake problem remains. The FlyWire Atlas is based on one female fly, with comparisons to a male and a partial hemibrain from another individual. Connectomics has shown that while the main highways are identical between flies, the small back alleys, the precise number of synapses between two neurons, vary significantly. This variability is likely where personality and memory reside. A fly that has learned to avoid a specific smell likely has different synaptic weights than a naive fly. The Connectome Atlas is the hardware manual. The software is written by experience. The mapping of the Drosophila brain is comparable to the sequencing of the human genome. In 2003, we got the list of genes. It took another 2 decades to understand what most of them did. Today, we have the map of the brain. The next decades will be spent exploring it. The FlyWire consortium is already pivoting to the next challenges. A complete map of the male fly brain is underway to understand the neural basis of sex differences. And looming on the horizon is the mouse brain, a structure 1,000 times larger than the flies. A mouse connectome would require zettabytes of data, a scale that demands new generations of AI and storage. But for now, we celebrate the fly. That tiny speck buzzing around your fruit bowl is no longer an annoyance. It is a miracle of biological engineering, now laid bare for all to see. Its mind has been decoded, and in its intricate wiring, we see the first blueprints of our own. Thanks for listening to G Fun Facts Online.